how important blood flow, and what is the biggest factor affecting blood flow. And obviously the biggest factor affecting blood flow is this radius. So the radius is to the fourth power, meaning that any small changes in radius is going to mean huge changes in terms of flow. One other thing right here, when we're looking at blood pressures, and I know we kind of mentioned it before, remember that there should be no sound at all, no sound, when you have a complete open vessel. It is only when you have a restricted vessel, whether it's due to a blood clot there, whether it's due to through an atherosclerotic plaque, right? Or whether it's due to a blood pressure cuff. Anything that can hinder and slow down movement of blood will allow us to hear now those carot cough sounds. So when you first pump up the blood pressure to 160 and you completely constrict the brachial artery, there's no blood flow. Because there's no blood flow, it also makes sense, you don't hear anything because there's nothing going on. As soon as the pressure in the cuff is less than the pressure in the brachial artery. So let's say my pressure in the brachial artery is 120 millimeters systolic. When the cuff pressure is at 119, then there's a little bit of an opening. There's more pressure in my brachial artery than in the cuff. At that point, a little bit of the brachial artery opens up and you have a little bit of blood movement. But that blood that's moving is in a constricted artery, right? So you will hear our first sound, the carot cough sound. And then you will keep hearing that sound and keep hearing it because until you hit the diastolic blood pressure, there's still pressure on the artery, keeping it from being fully open. When the diastolic blood pressure is achieved, what the diastolic blood pressure measures is this. That is the blood pressure that we see when the heart is at rest. Now, that's not the answer that I'm going to get, that I'm expecting, because I know some of you guys haven't turned in that sheet yet. And I want it turned in by the end of the class today. So I'll give you guys time to actually look it up in, between breaks and stuff like that. But just keep in mind, it is the blood pressure that is from between, right, heartbeats. But it's not just that. Because between heartbeats, the pressure goes to 0 in the heart. And our diastolic blood pressure is not 0. It's something a little bit more subtle of an answer. So, the diastolic blood pressure, the artery is open, and now you have resumption of normal flow when there's actually no movement at all, there's no constricture at all. Because there's no constricture at all, you have equal movement of blood that's laminar, and what happens then? Well, what happens is it's normal flow now, there's no constriction. That is going to be an absence of sound. So the absence of sound, and it's always hardest to hear an absence of sound, right? Once you hear that carot cough sound, you hear it, you almost anticipate it. And when you don't hear it, you almost think like you're kind of losing your mind over it, right? Maybe I missed it. So yeah, it is when it is very subtle, that diastolic blood pressure measurement. Now I will give you the answer to this for diastolic blood pressure and MAP at the end of class today. So if I don't have your sheet in by the end of class, you're not going to get it. So now let's talk about in terms of physiology of the systemic circulation. The physiology is determined by the anatomy of the circulatory system, dynamics of blood flow, and regulatory mechanisms that control heart and blood vessels. Now, blood volume, and we kind of mentioned it, where do we find blood most commonly at rest? And this picture showed that at rest, most of the blood is in your systemic veins. Veins in the lower legs predominate as well. So most of the blood is found in the veins, and when you start to work out, we start mobilizing that blood that was trapped in the veins. And now it's like receiving more blood than previous. Now you have increased venous return, increased cardiac output. Now let's talk about this cross-sectional area and where we see the greatest velocity. The greatest velocity is in one artery that is very big. That's the aorta. Take a look at how fast blood flows as it goes through the aorta. Why? Because there's only one of it and it's very large. It's like you go to Atlanta and there's like 10, right? 10 lanes in one direction and there's no traffic at all. You're just gonna go super fast, right? And that's what's happening in the aorta here. It's super big, and now there is no exit or entrance ramp. It's just going straight ahead. Once you're in, you go straight through. Why is that important is once we get into the smaller arteries and arterioles, we're gonna split into many different sections and have many different exit points here. And when you hit those exit points, your velocity is going to slow down. And that's what we see as we go from the aorta, one aorta, to the muscular arteries. And then the biggest slowdown we see is what happens when we go from blood going into the muscular arteries into the arterioles. Look at that huge decrease in velocity. Now, why do we see such a huge decrease in velocity? It's because as we go into the arterial level, you can have many, many arterioles rising from one muscular artery. So you can have lots of branch points. At any branch point, and every branch point, blood is going to slow down at those branch points. So if you think about it, right? When you are driving and you have exits on each side, and if you go to Chicago, you'll have that. You'll have an exit to the left, exit to the right when you're on the freeway. It really slows everybody down, and you can get traffic jams like that because people who are not used to driving there don't know that you should be on the left side when they exit, or they should be on the right side. So they're crisscrossing, changing lanes, so then they can hit their exit. So the more exit points, the slower things move. And that's what we'll see here. So the greater the exit points, the number of exit points, the slower the velocity. Now keep in mind, we want a very, very, very small velocity, right? The slower it moves, the more time we will see for filling, for gas exchange, for nutrient exchange, for waste exchange. So in the areas where we do have gas and nutrient exchange in the capillaries, it kind of makes sense that that's where we see the slowest, right? That's where it goes pretty slowly, so then there's time for each red blood cell to go from one end of the capillary to the other. Now, the greater the cross-sectional area, the slower the speed. That's the big thing about this slide. So in terms of the aorta, it's big, but there's only one of them. So in total cross-sectional size, it's pretty small compared to how many capillaries we have. The capillaries, each one is microscopic, but you can have billions of them. And when you multiply that microscopic capillary by billion, you're gonna have a huge cross-sectional size for the capillaries. So here, aorta, 5 centimeters squared only, whereas in the capillaries, 2,500 millimeters squared. So that's pretty crazy, centimeters squared. This low rate of movement of blood in the capillaries allows for gas exchange and nutrient exchange. Pressure and resistance, and here's another example of this. I want, for these 2 slides, there's a couple things I want you to know. One is there's that drop in velocity. That drop in velocity in the arteriole slows down blood movement in the capillaries. That drop in blood in that movement right there, also in the arterioles, we see the greatest drop in pressure. So this is our systolic blood pressure at 120. Our diastolic at 80. The mean blood pressure is an average, right? So what we see is that there's a huge decrease when we hit the area of your arterioles. From aorta to the end of our arteries, the mean blood pressure in the aorta, 100 millimeters of mercury. When it hits your arteries, we're still above 80. And even when it enters the arterioles, we're still above 80. But once it enters the arterioles, right here, When it enters the arterioles, it's about 85 millimeters of mercury. When it exits the arterioles, it's at 35. There's a massive drop in pressure as we go from the muscular arteries to the arterioles. Now, why? Well, because arterioles need to kind of control the amount of blood that goes into the capillaries. If we allow too much blood into capillaries with too much pressure, we will blow out those capillaries. Those capillaries are microscopic, they're thinly walled, and they're basically one cell thin. And if we allow a lot of blood with a lot of pressure, we will burst those capillaries. So the arteriole's main job is to decrease the pressure of the blood as it enters the capillary, as it's driven from one side of the capillary to the other. Again, greatest drop in pressure occurs in the arterioles, which then regulates blood flow through the tissues. No large fluctuations in the capillaries. So in the capillaries, you're at about 30. And then when you exit the capillaries, right here in the venules, you're about 15. So there's some drop, but not massive. Like here, we saw like a 50 millimeter drop, right? It was at 85 going into arterioles. As it exits arterioles at 35. That's 50 millimeter drop. That's huge. Muscular arteries and arterioles are capable of constricting or dilating in response to autonomic and hormonal stimulation. Muscular arteries regulates blood flow into the specific region of the body, and then the arterioles regulate flow into a specific region of an organ. So the muscular arteries are going to direct it, so then if you're working out, you're going to detour it from the celiac trunk, from the renal arteries. So then you can keep that blood and all of its nutrients for the skeletal muscles of the legs. And the arterioles there, right? The arterioles in the GI tract also constrict. So whatever blood that goes through, actually you have a constricted area, so then you detour it even more. Again, so then you have minimal amount of blood going into regions that are not active. All right, so pulse pressure, diastolic minus systolic minus diastolic blood pressures. Difference between systolic and diastolic pressures increases when stroke volume increases or vascular compliance decreases. We're not going to use, so with the pulse pressure, just ways of palpating for a pulse. And if you take a look at it, these are all the different regions where we can palpate for a pulse. Again, there's a difference between palpating and feeling and auscultating and hearing. None of these should be heard, but they all should be felt. Now comes the bulk capillary exchange. Now this is going to be, when we start talking about this area right here, this is going to be different than what we're going to see with nutrient exchange and waste exchange. In terms of capillary exchange, the most important means of exchange is by way of diffusion. Lipid-soluble molecules cross the capillary walls, diffusing through the plasma membrane of our simple squamous epithelium. Again, one of the things that we worked about last semester was figuring out why things should make sense. Why should it make sense that we have simple squamous epithelial cells forming the capillaries? Well, it makes sense because simple squamous means it's very flat. Because those cells are very flat, it's much easier for gases to go through those cells. From inside, where the red blood cells are carrying the oxygen, then the oxygen can go through the cell, go to your target tissue. If you have a cell that's simple cuboidal forming these capillaries, well, simple cuboidal is thick. It's going to be very hard for that diffusion to occur. It's a much thicker cell. So you're not going to have, hopefully, those cells in that area where you need simple diffusion. We want small, thin, flat cells. And that's what we see here. They allow for diffusion. Lipid-soluble molecules cross the capillary walls and diffuse right through the plasma membrane into the tissues that it supplies. Now, what are these lipid-soluble molecules? Obviously, steroids, right? Now, we think about steroids, right away everybody thinks of anabolic steroids. Those are not the steroids we're talking about. testosterone, estrogen, progesterone. Those are some of the steroids we see in our body. But we also see, besides those gases, goes right through without any help. And that's good, because the last thing we want to do is wait for a transporter for oxygen to get into your cell. We want oxygen to just go right through, right? And that's what we see. Carbon dioxide as well. Again, why do we have so much carbon dioxide? And we're going to talk about it when we mention our Krebs cycle. Remember, so when we look at something like glucose, glucose is just nothing more than carbons with lots of oxygens they're attached to. Right? So right here, you have glucose. Well, is the structure of glucose important? Not really. What's important about it are the electrons forming the bonds. What happens to all of these carbons and oxygens? They turn into carbon dioxide and water. That's what they turn into, waste, right? So all these elements, all these atoms that we define right here, that makes, right? What's important about them are the electrons that are forming bonds between them. It's a spark that we use so that we can make ATP. The rest of it is just waste. Now, because we're using glucose a lot and using oxygen a lot, we're going to be producing a lot of carbon dioxide and water. We need it to be soluble, to go in and out easily without any help. Water-soluble things diffuse through the fenestra or the intercellular spaces, or need a little receptors to help get it through. Glucose has this, we have receptors that can help move it through the cell. Blood pressure, capillary permeability, and osmosis affects the movement of fluid from the capillaries. Fluid moves out at the capillary at the arterial end. Most, but not all of the fluid returns at the venous end of the capillary. That remain in the tissues is picked up by the lymphatic system, then later returned to the venous circulation. Now, what does this mean right here? This means this slide right here. And usually I write it and I draw it just kind of differently. And what we're going to see, maybe I'll write it over here, is this. When we take a look at this slide and take a look at this, it's going to be another picture of multiple tubes, just a little warning, right? That's the way I draw. So what we're going to see when we look at the movement is what we call fluid exchange across the capillary. This is different than diffusion, right? This occurs with diffusion. So you have diffusion of gases, diffusion of glucose in the capillaries. That's simple diffusion and facilitated diffusion. This is a different other kind of process besides that. So let's think about what happens here. So here's an arterial end. So here's the arterial end of our vessel. Over here is our venial end. So remember, they're connected, right? It's one closed system. Artery moves blood into the arteriole, which moves blood into the capillaries, which then the capillaries are connected to the venules, right? So it's one closed system. So we have blood moving through the arteriole, and the arteriole is going to be connected then to the venule. Now I'm not going to write and draw a capillary bed, but what we're going to see is that this is our capillary right here, made out of simple squamish epithelial cells. This area is our target tissue. The tissue we're trying to bring nutrients to. Nutrients like oxygen from red blood cells, they just pass right through. Now, why do they pass through though? Why does oxygen go from your bloodstream to your tissue? Simple, what we just mentioned, right? If we're using aerobic respiration, we're going to be using a lot of glucose and a lot of oxygen. If we're using a lot of oxygen, it makes sense inside my cell, I'm gonna have less oxygen. I'm using it all, right? So inside my cells that are active, I'm gonna have lower oxygen level. Now, oxygen is a gas. So with gases, we can't just say, you have lower amount of oxygen per mole, right? Instead, we use, for gas, another symbol, P. PO2 and PO2 over here. Partial pressure, or P, or pressure in general, is a way of measuring the concentration of a gas. You can't see a gas. How can you tell when you have a certain amount of gas? Well, gas exerts a force on us. So when we feel that force, that's pressure from that gas. So when we talk about the partial pressure of oxygen, it is a concentration of oxygen, right? That's how I want you to think about it. In reality, right, you can't see it. All we can do is feel its effect on us. And we feel its effect by the force or the pressure it exerts on us. The greater amount of oxygen, the greater the force it hits us with, right? The greater the pressure we feel from it. So if we're active, our oxygen levels will be low. Low oxygen here, very high in my bloodstream. What happens? Diffusion goes through, right? And if we're active, what else do we see? High CO2 inside my cells, because I'm going to produce a lot more waste. So high CO2, low CO2 in my bloodstream, gas goes PCO2, or CO2 goes in reverse. This is why those gases move in certain directions. Oxygen always goes out to the tissues. Why? Well, the more active the tissue, the more oxygen it uses. Now we have a concentration gradient for oxygen. So then oxygen wants to go into your cells because of that low oxygen content. The opposite is true with carbon dioxide. In the process of forming ATP, I'm going to be forming a lot of carbon dioxide as waste from glucose metabolism. So I'm going to have a lot of carbon dioxide in my cells. Now I have a concentration gradient for that carbon dioxide. Carbon dioxide wants to go from your cells, and it wants to go to an area of lower concentration, which is in your plasma, which is in your bloodstream. Perfect. Exactly what you should see. That is just diffusion. What we're talking about here is about movement of liquids now. Now, if you remember on the arteriolar end right here, we have a drop in pressure from 85 millimeters at the very start to 35 millimeters of pressure when we hit the capillaries. So the blood pressure, as blood goes into the capillary here, the blood pressure equals 35 millimeters of mercury. And that is what they call our blood hydrostatic pressure. Basically, your blood pressure, right? So on one end, because of blood pressure, you're forcing blood with a lot of force to go into the capillaries. And now you have tiny little gaps between those capillaries. What happens then? Well, what happens is, it's like our garden hose with tiny holes in there. As I start turning on the water spigot, right? When I first put in just a little bit of water, a little bit of water flows through my garden hose, you have a little bit of leakage out, and that's it. But what happens when I turn the garden hose, the spigot completely on? And I have a lot of flow, right? As you have a lot of water moving through the garden hose, you create a force, and you cause the actual garden hose to swell up. Any tiny gaps, well, water is going to leak out through those gaps. And that's what we see here. In the arterial end of a capillary, because of the higher blood pressure, we're going to filter, push out water coming out from the bloodstream. Now, red blood cells, they are too big. They can't get through those gaps right here. Albumins, proteins, they're too big. Can't get through those tiny gaps between the cells. But water can seep through. And they seep through because of these tiny gaps allows water to get through. Now, keep in mind, imagine there's a garden hose again, and now you've turned that water completely on. You have a lot of water going through your garden hose, and there are holes everywhere in a garden hose. Nowadays, they sell garden hoses with holes in there so that you can spread out the water, right? So what happens? The more holes, the more water that is lost. That's what we see here. So water is lost, and we're gonna see filtration occurring on the arterial end of our capillary. They say 33 millimeters of mercury, I wrote 35, right? So about in the mid-30s, right? So you have this driving force because our arteriolar pressure is higher on the arterial side, 33 to 35 millimeters of mercury. But what happens then? If I keep adding water and pushing it out, well, I'm gonna have some water that comes back inwards, right? Because if I keep pushing water outwards, osmotically, water wants to come back in. Because I keep putting water into the interstitial space here, what happens? I'm gonna start diluting that water a little bit. And some water that I just pushed out gets sucked back in. That accounts for the 20 millimeters that we reabsorb right away. Total, we have about 13 to 15 millimeters of water that we push out on the arterial end. Again, all right, this is different than what we saw with filtration, with diffusion. Here, we're gonna see in the capillaries, on the arterial end of the capillaries, we filter more by pushing out more water at a rate of 13 to 15 millimeters of water. Now, what occurs as we move downwards towards the venule? As the blood goes through the capillaries, that blood is going to then enter a venule. As it enters a venule, what happens? In the venule end right here, the pressure drops. It was at 35. Now it's going to drop to 13 millimeters of mercury. That's our blood hydrostatic pressure. What else do we see? We have a reabsorption pressure due to osmosis. And again, right, water wants to be drawn back in. Because I keep pumping so much water on this end, what happens is I dilute the water in the interstitial fluid. Water wants to go through by osmosis and go into the bloodstream again. Before, what we saw was we're pushing that water out because of hydrostatic pressure. But as blood goes from one side to the other, the blood pressure drops from 35 to 13. Our osmotic pressure stays the same, 20 millimeters of mercury. Osmotic pressure. What we see is on the venule side, we see anywhere from 7 to 10 millimeters of negative oxygen or negative water pressure, meaning that on the venule end of a capillary, we have a lot more reabsorption of water. And this is what allows most of the water that we pushed out to come back in. Think of this effect as a suctioning effect, right? You pushed out all that water, and as that blood goes from one side to the other side, the venule side, the blood pressure just drops. As it starts dropping, right, a suctioning effect occurs where water just comes back into the capillary. As water starts to come back in, it makes sure that we don't lose so much of it and we have edema everywhere. Right? Now, this is going to be very important when we start talking about the kidneys. The kidneys use this concept for filtration. On one end of the kidneys, we're going to be pushing a lot of water out. So then that water can be going through the kidney tubules. We can reabsorb it all. Right? At the same time, we don't want to reabsorb all the water. If we do, we can't get rid of some of the waste we make. Urea is some of the very, very important waste product we make. And in order to get rid of it, you have to form urine. If you don't form any urine, and you guys have probably heard that if you don't form urine, it's a bad sign, right? It's a bad sign if you don't form urine because it means you're incredibly dehydrated. And at the same time, now you can't get rid of the waste at all. And it's going to start being maintained inside your body, and it's going to build up inside your body. So forming a little bit of urine is required for us to get rid of waste, like urea. You're going to see, this is how we do it, right? We have blood vessels just like this in the kidneys. And in the kidneys, we're going to filter more on one side than we reabsorb. There's always going to be a lot more filtration than reabsorption in the kidneys. And even in our normal tissues, like your arms, right? Your blood vessels in your arm, your legs, we're going to have more filtration, 13 to 15 millimeters of mercury of filtration, only 7 millimeters of mercury for reabsorption. So there's that a little bit more filtration than reabsorption. So meaning we're going to always have a little bit of water that we push out into the interstitial fluid. By the way, it's hard to imagine what the interstitial fluid is. I want you to think, you guys have gotten a cut before, and when you get that cut, does it look dry? No, it looks pretty wet, right? When you look at it, no matter where you get a cut, it's got that wet, shiny look to it. That wet, shiny look is because of fluid there. That's an interstitial fluid. Or sometimes you get a cut and there's like that yellow fluid that comes out. That's interstitial fluid that we see in our body. And that's found throughout our whole body, right? And that interstitial fluid is formed by this filtration system. So then we always leave a little bit of water because we always filter a little bit more than we reabsorb. Now, under normal circumstances, most of this excess water we filtered, under normal circumstances, most of it will be returned by way of your lymphatic system. So we have another system that kind of allows us to return some of that excess water. So then we have enough for the interstitial fluid. At the same time, you don't keep getting bigger and become edematous, right? We have the lymphatic system that takes this excess amount of water we pushed out. And that lymphatic system allows us to then reclaim that water, that fluid, and reincorporate it into our plasma by introducing it into your subclavian veins. Now, we'll talk about it later on as well, but there are things that does happen that allows us to filter more or reabsorb less. And it can cause people to have huge problems like lymphedema, right? Elephantiasis, you guys have probably heard of that. Probably from movies, they talk about elephantiasis. Well, elephantiasis, and it's not called elephantitis, it's elephantiasis, is when you have the legs become huge. I did a medical mission work. I went medical missionary work to like 3 different places, and none of them were really that great, right? So, I mean, in terms of like, you know, we were in the bush areas of Africa. So I was in Mali and Burkina Faso for 2 months. Right before, right, we were about to, again, for a 3rd month, we're about to go to the Ivory Coast, right, Cote d'Ivoire. And then all hell broke loose right before we were about to get there. They had the start of the Civil War that lasted like 10 years. So we're lucky we didn't get there, but you know, you can imagine when you're going in those areas, if you're stuck there, you're really stuck there. It took us to go from the capital of Burkina Faso, Ouagadougou, right? It took us 3 hours to go from the capital to about 50 miles away, right? And we had brand new trucks, right? We had brand new equipment. And it took us almost 3 hours to go about 45 miles. Why? Because the bush areas were so far away. There, we saw a lot of people that actually had elephantiasis of the legs. And there's nothing you could do. The legs were like, one leg looks like tree trunks, just huge. And there's nothing you can do to alleviate the stress on that body. It's caused by the parasite that goes into the lymphatic system, a fluke that goes there. As you trap and kind of make the lymphatic system become not useful, not functional, now that excess amount of fluid here starts to build up every second of every minute of every day. And then it causes your legs to swell, right? and it keeps being swollen and swollen and swollen, and it keeps getting bigger. And then, right, at that point when it gets big, there's really no good treatment plan. They've tried debulking it, removing some of the fluid and some of the tissue there, and it just grows right back. Again, important thing to note about this is just understanding that in order for that this kind of process here, this kind of filtration on one side, reabsorption on the other, we don't normally see it occurring in us that are healthy because we don't have a buildup, right? We always have the lymphatic system functional, so then we reincorporate whatever we didn't reabsorb right away. We are able to have that fluid go through the lymphatic system and we reintroduce it into our bloodstream. Now, we do see it when people have other issues. If you've had mastectomy and you've had a sentinel node biopsies, or you've had all of your nodes being removed, what happens then? Well, you can't get that excess fluid back to the bloodstream. And then anything can cause you to have a little bit of lymphedema in your arm, right? And if you do blood pressure cuffing on that arm, right, it will become edematous and huge for months at a time. Very painful. And all it takes one time for somebody to make that mistake. And then the patient will be like, no, you're not gonna do blood pressure on my arm now, right? Again, if you can't do blood pressure, if you have double mastectomy, what do you do then? If they're not gonna let you touch the arms, what do you do? How do you take their blood pressure? The neck? No, don't do the neck. It's not gonna, it's just a joke, don't do the neck, right? The femoral. But the problem is, the femoral artery is not at the same level as your heart, right?
Put your leg up, right?
Right? Not put your leg up, no. You have them lie down flat,
Okay.
right? You have them lie down flat. If you're laying down flat in a prone position or in a supine position, doesn't matter, your heart and your legs are at the same level. So you shouldn't have an artificially higher blood pressure as a result of it. Questions so far, guys? And again, we shouldn't see, in terms of this, it shouldn't be like a massive amount of fluid that we see that is net filtered, right, and not reabsorbed. It shouldn't be like that. Here we should see that it's only